Hello, I'm Will. Welcome to ResearchPod. In market terms, pharmaceuticals and biologic companies represent a growth opportunity like few others. Vaccines alone have had over 350% revenue growth between 2007 and 2018, well before the COVID-19 pandemic. So how can a researcher behind the bench today think about broaching business? Professor Vivek Kumar from the New Jersey Institute of Technology is an advocate for, and success story of, developing companies from a research backing. We talk today about funding, finding those angles to commercialize your research, and what he sees as key developments that could revolutionize both business and technology. Vivek, hello.
Hello, sir.
Thanks very much for your time in joining us today. For my own information and for everyone listening at home, could you tell us a bit about yourself, some of your personal and academic background, and how you are managing to kind of bridge the worlds of academia and business all at once?
Definitely. Again, my name is Vivek Kumar. I am an associate professor of biomedical engineering at the New Jersey Institute of Technology. And here at NJIT, in my research lab, we innovate new biomaterials-based drugs. So what's unique and interesting about this is that these materials that we make out of proteins, these materials that we make activate cell receptors, so they're classified as drugs. Anyway, so part of doing all of this is encouraging young minds to come up with these neat ideas to regenerate tissue, treat different diseases. We even had a therapeutic against SARS-CoV-2. But another aspect of that is you come up with these ideas. How do you take them to market? Not just market, how do you get them to treat someone? Like the dream of someone like me who went to school for bioengineering undergrad, bioengineering graduate school, got my PhD. I went to Northwestern undergrad, Georgia Tech. For graduate school, I worked with a surgeon, Elliot Chaikoff, who then moves up to Beth Israel Deakin as part of Harvard Medical School, where I finished up my PhD, did a short postdoctoral stint there. And then at Rice University, I did another postdoc where I got into protein engineering. Anyway, the dream of someone like me, a biomedical engineer, is to take something we've come up with, we've invented, and put it into human beings, to treat some disease in human beings. And as a part of that, we invent these technologies, file intellectual property, patents, and things like that, publish papers, our currency, right? Papers and grants in academia, but we try to translate them using startups, using seed stage, small idea-based companies. And one of the challenges with this is how do you get money? How do you get people to run these high-tech, complex engineering projects and try to gain traction? So it's been a tremendous adventure over the past 6 years as a professor and 10 years before that training to get to this stage doing research. But it's been a really interesting adventure on how we can, develop ideas and make them into seed stage startups and go out there and pitch, try to get venture capital investment or angel investment with the many hats I wear to move technologies towards the clinic. And I think one of the greatest things and one of the greatest experiences in all of this is you learn so many ways to fail, both from academia all the way to pitching, to VCs, to angel investors. And as a part of that, you really learn how healthcare and medicine is practiced, how healthcare technologies gets implemented and taken towards the market, towards the clinic. And there's so many factors, so many push and pull factors that you couldn't even imagine influence how much uptake there is for a drug or a therapeutic, which has been a fun challenge, but learning experience as well.
Yes, I mean, you highlighted some of the many steps just in the academic development, let alone, and then thinking, actually, there might be some business, there might be some money in this, and picking that up as a profession. I suppose to look back on all of that, if you were to be able to send a message back to your younger self and tell, a fresh postgraduate you one thing, would it be about the business or about the academic sides, or do you think it would even be prepared for everything that you've undertaken since that first degree, let alone all the ones that followed?
So I think, the bachelor's I did, my PhD, the postdoctoral experience, all of those were building blocks, right? Like I always think back to myself, why did I have to learn calculus and linear algebra and all those things back in my high school and college? I never used that in my daily life. To be fair, every now and then I do. And it's all these tools and the struggles going through those that I have learned the resilience and perseverance to take that next step. Also, it's things that you might use and it's important tools to have to approach these different problems. If I could go back in time, I would tell myself what I tell all the students I advise today, which is go out and explore. I don't think I made the mistake of not exploring enough. I think I was a little scatterbrained in my approach. I think I could have been more focused in my approach of exploration. I know it sounds somewhat, oxymoronic, right? Focus exploration. But I tell my students, if you want to be a physician, if you want to get a PhD, if you want to go into healthcare or any different aspect of any career, go speak to someone who does it every day, right? Go speak to a clinician, not just a surgeon, not just a PCP, primary care physician or GP. Go speak to a psychiatrist. Go speak to someone who works with pediatrics. Go speak to someone who works in, underserved populations. Go speak to a private clinic. Get a full range of perspectives. Because I'll tell you this, one of the lessons I learned by speaking not just to physicians, but speaking to patients who have debilitating blindness, right? A condition called wet age-related macular degeneration, diabetic retinopathy as well. One of the leading causes of blindness in people above the age of 55. Too many blood vessels on the retina. You inject drugs to kill the blood vessels. The biggest challenge is not injecting drugs into the eye. No, because they're used to that. The biggest problem for those patients, these elderly patients, is inconveniencing their son or daughter to take them to the clinic because they can't drive there and they can't drive back. So they have to take time out of their children's lives to usher them to the clinic. And to figure something out for that, that's a big innovation. So our goal there was to figure out, can we develop a long-term release solution for 6 months instead of monthly injections, right? That is the key innovation. It's not, can we find a better anti-angiogenic drug? It's not, can we find a new molecular target? No. It is, can we ease the burden of the patient population? And those are lessons that you cannot learn unless you have conversations with people in the field that you hope to get into. So if you want to be an ophthalmologist, go speak to someone who's suffering from blindness due to diabetic retinopathy. Go speak to a patient advocate. Go speak to the clinician. Go speak to the pharmacy benefits manager. Long story short, if I could go back in time, I tell myself to explore and learn as many different aspects of a field as possible, because otherwise, you're just not making very informed decisions.
Well, you mentioned the lessons that you pass on to your students there, and that, I suppose, all comes under the umbrella of the Kumar Lab. Could you give us maybe a quick tour of what happens there, the developments that go into not just the drug design and devices, but the tissue engineering, the protein engineering that you mentioned, and I suppose the teaching materials and the teaching experience within that lab?
So in addition to doing research, I teach as well. I teach an undergraduate advanced biomaterials class. And in that class, we talk about different applications of materials. Specific to my research lab, where I spend most of my time in terms of advising my students, we do drug design. We look at novel receptors. We use complex computer programs. Some of the gaming computers that gamers love, we buy those because they run computational simulations really, really well. Graphics cards, GPU power, right? Graphical processing unit power. So we have some pretty cool computers in the lab where we do drug design all the way to synthesizing them. We have peptide synthesizers. So there's a lot of safety training we do in our lab to make sure students are safe with the different equipment we use. But also, I'd like to think we train translational scientists, right? So not only do we synthesize materials that make scaffolds that we can implant, look at tissue processes, look at localized drug release, localized tissue regeneration, wound healing, stuff like that. Not just that, but also teaching this idea of, hey, if you have an idea, if you want to treat a certain kind of disease that doesn't have a treatment right now, what is the gap in the field, right? Let's say there's this new receptor that no one has targeted. That's a gap, right? Let's figure out how to target that. So all the way from computational design, ideation, to implementation, to doing efficacy studies in rodents, in canines, in small, large animal models, in vitro, in petri dishes, synthesis, characterization, materials generation, testing. What I hope to do with my students is train them in this full gamut of being a translational scientist, having the tools, skills, ability, and maybe even getting their hands wet in all the way from ideation, computational peptide design in silico on a computer, all the way to making it and testing it to see whether what you came up with actually works. And that's something that we're very excited about. And each and every one of those, because it's new intellectual property, we try to file patents and we try to get novel claims on what these intelligently designed, novelly designed materials can do.
Now, I'll come back to the kind of delineations between biology and business in a second. But it sounds like straddling the world of biology, business, but also what a lot of people are going to be hearing about, if not immediately, then very soon, about massive data processing that can be done with artificial intelligence, machine learning, that, coming up with chat GPT prompts to come up with a new story or to come up with any text-based ideas, that's one thing. But to then be able to scattergun approach 1,001 different atomic combinations, 1,001 different molecules at once, like you're straddling not just the two worlds of biology and business there, but computational science as well. Does it ever get tiring having to learn something new about the bleeding edge of everything every day?
Oh my God, no. So coming back to exactly the question you asked me at the beginning, right? It is a toolbox that every day is growing bigger and is being enriched. And I cannot think of a better time to be doing science than today and than yesterday, right? Because ChatGPT is great. ChatGPT, ChatGPT-4, what have you, is a great tool to start asking some questions. Now, the complexity of those questions starts to get to a point where you need quantum computing. So not only am I excited about AI and ML, artificial intelligence and machine learning, to help me think about how to approach these problems, but quantum computing can now help answer those problems. And let me explain. Your classical computer, the computer we're talking about, right? The computer you use every day, your phone, what have you, it runs data on logic gates on your typical classical computing. And it works, it does calculations pretty quickly. But when you have to guess what kind of peptide protein sequence binds to a receptor, each and every peptide has 20, in general, commonly occurring, 20 possible combinations. If you have a 10 peptide long sequence, it's 10 to the 20 combinations. More stars in the universe, right? So 10 to the 20 combinations, which will take you till the end of time to start guessing if all of them fit. So now you've got to use some kind of computational power to start guessing these ridiculous numbers, which I don't know the word for, right? Combinations. You cannot do that with a classical computer. You can do slightly better with those GPUs that I was telling you about, right? But with a quantum computer, you can start doing these insane calculations. You can start calculating these numbers, combinations, which are so intractable, right? One of the same reasons that when people first started talking about quantum computing, they're like, oh no, security is in jeopardy because now you can guess any security key because there are so many combinations, but this can guess them, right? In the same way, quantum computers could solve all the Bitcoins out there in like a couple of hours as opposed to 40 years. You know what I mean? Anyway, I'm getting lost in that space. The point I'm trying to make is as IBM, as Google, as all these companies are making 128, 250, whatever bit quantum computers, once we get quantum computing up the scale, we can use the algorithms in AI and ML to start capitalizing on quantum computing to guess better what will fit a receptor. Long story short, we can make designer drugs and designer materials, my focus materials, very, very quickly, very, very efficiently in the next 5 to 10 years. That's going to revolutionize medicine.
Well, I think that the potential there and the partnerships that are going to be required to make that potential become a reality, hopefully along the kind of lines that we're seeing here, and not just, liquid metal T1000s coming to kill us all in our sleep or something. Like, that is very recently, well, until recently, was sci-fi. It's now bleeding edge stuff that could materialize over the next 5 to 10 years. For people who have been working in universities, in labs for the last maybe 10, 20 years, that's going to seem possibly big and scary as a change. And like doing anything that is not the lab work in front of you can be a big and scary change. And it sounds like you have been embracing big and scary change every step along the way.
Well, COVID taught me that, right? So when COVID happened 3 years ago in March, April of 2020, I was told my lab has to shut down, as was everybody else. Everybody had to shut down. So the only thing we could do is one of two things: either work on COVID, right? Because then you can get people in the lab to work, or do computational work, right? I mean, there's many things you can do, but those are two big things that folks like me could do, right? So we did both. So we came up with an idea. So I told you about proteins. We work on proteins. One of the proteins in your body is collagen. And the way collagen works is your cell makes collagen and it self-assembles into a rope, into a fiber. That fiber self-assembles into tissue, right? You get very strong strength, bone, skin, everything, because proteins self-assemble. They come together on their own, like collagen. Our proteins self-assemble as well. So our peptides form beta sheets they form antiparallel beta sheets, they form fibers, and they form hydrogels. So with self-assembly, we ask the question, can we bind to the virus? Because the virus has something called spike. Can we bind to spike? And can we self-assemble on top of the virus, preventing it from interacting with human cells? So we came up with that idea. We filed a patent. We applied for a grant. We got some funding. So that's one thing that we did in the research lab. We're trying to publish that work now. In addition to that, what we've also been looking at is, ever since COVID came about, is maybe 25 to 30% of my lab has transitioned from just wet lab and animal work to computational work. And we do a lot of computational simulations ever since then, and we've started to publish in that space as well. So yeah, I think if you do not embrace the latest and greatest technologies, you end up doing what they did 50 years ago or 100 years ago or even 10 years ago, which is go out into the Amazon rainforest, go out into many different places and do random screens of bunches of different chemicals and plants and different distillates and things like that, and try to figure out, oh, this might work, but you don't know the side effects. Now, with much more rationalized design, with the technologies we have today, we can see with almost molecular scale or almost atomic scale resolution the structure of proteins. And then we can design other proteins with, again, near atomic scale precision to bind them and target them, right? We're getting to a point where we can treat many, many diseases at the near atomic scale of precision.
This is making me miss leaving the lab. I mean, like you talk about spike protein. I have a beta sheet tattooed on my arm. They were simpler times, but this was more fun.
But I have to say, like, you always have people who are excited and enthusiastic about these things. But at the end of the day, there are still so many challenges. One of which is delivery. How do you get it to where you want it to go? And for it to stay there, stick around, last a long time, do what you want it to do. The body is highly heterogeneous, trillions of cells that are all trying to do a variety of different things to break stuff down or what have you. Immune responses. And then, of course, that's the academic or biological side. Then you've got the funding side. If big pharma, med device, what have you, is not interested in that portfolio of products, doesn't have interest in that, taking that technology forward, it's very difficult to justify clinical trial costs, which cost hundreds of millions, if not, tens of millions to hundreds of millions of dollars to do. So there needs to be a strong justification and understanding of pharma or med device appetite, even before starting something, in my opinion. Understanding the whole translational gamut is critical in every step of the design process.
Well, we talked about some of the kind of the practical hybridization that you've been doing between wet and computational science. To think about the philosophical hybridization between biology and business or, commerce, market development. When, I suppose, was the first time that you had the first notion about commercializing a model? When kind of in your personal trajectory was that? And you mentioned that you've learned a lot from failure, so was it a success? And then how long until the first big success came after that first idea?
Right. So I guess when I was doing my postdoc back in end 2012 to early 2016, I was at Rice University and I was doing self-assembling peptides. We're working on angiogenic peptides that help generate blood vessels. And I was a scientist, right? I love doing science, publishing papers. I didn't really care for the business side of things. And I thought to myself, I'll let the businessmen figure that out. I'm happy with science. And one of the patents that I had filed at Rice University, along with my advisor, Jeffrey Harkering, was taken in to be a part of this entrepreneurship course because they were looking for technologies. And it just so happened that I was brought in as an advisor for that course. I ended up taking the course anyway. And long story short, through that course, I started realizing there is so much more to medical device and drug development than sitting behind a research bench. And it is easy to figure that out. The easiest way, go speak to someone who actually does it. Go speak to someone in pharma. Go speak to a physician who administers the drug. Go speak to a pharmacy benefits manager who manages what drugs a formulary or what drugs a hospital or clinic buys. Right? Go speak to different people in this ecosystem and actually understand that what you're doing at the bench may end up in Nature, may end up in these great journals, but may never go further than that. What really matters is you had this perspective on what is required for translation. So I do this course, I realize, sure, basic science is important, but translating it, coming up with lessons, understanding the pushes and pulls on what drives translation, what drives these ideas forward is important. And that transformed the way I do work. I no longer ask questions for the sake of asking questions. Every question I ask in the research I do is translationally motivated. Like, how can we improve delivery for this target, for this application? I believe, and I think that segues perfectly into the business side of things or the entrepreneurship side of things, in that if you have the end target in mind, if you understand that pharma requires X, Y, and Z, this is the kind of efficacy study you need to do, as opposed to just vanity science, right? I think answering those questions is a lot more important and a lot more prudent use of taxpayer money, right? In terms of grants and things like that. Coming up with strategies, technologies that benefit and help human society. A lot of failures along the way. In fact, my first company, Nanjotex, is still going right now. We're trying to develop an angiogenic hydrogel for dental pulp regeneration. We've had many hiccups along the way. One of the lessons that I think one can learn throughout this process is pivoting. And also, you only learn through failure. Almost everything I know today in terms of entrepreneurship, in terms of even self-assembling peptides, the applications of the materials I use, I've learned after my formal training in school, for example, after my PhD, after undergrad and all of that. Sure, I needed to know Argo and transport and things like that I learned in college and my PhD. But what I do today, I've learned after. Skills I've learned on the jobs, skills I've learned by going out and pitching at events and winning or losing or getting someone coming and telling me that it was terrible or great. So,
do you find that just the time commitment in any one day of balancing a teaching commitment versus lab time versus attending a board meeting for one of the companies is a juggling act, or do things just kind of take the time that they take?
So the companies that I have are very small in that they have either 1, 2, or 0 employees outside of myself. These are C-stage startups. A lot of them, what we try to do is we apply for federal grants or state grants, different kinds of funding initiatives to get pilot seed money. And then we apply for bigger grants or go out to apply for VC, angel money, things like that. Oftentimes at a seed stage startup that's doing pharmaceutical development, med device development, most of the time you're not selling to the market. Most of the time you will exit to pharma. You'll exit to a bigger partner once you're in late stage animal trials, late stage clinical trial, or early stage, late stage, mid stage clinical trials, because you don't have the infrastructure, distribution network, things like that to compete with big pharma. However, if you partner with them, you can synergize and translate that way. And that's the goal of my companies as well, is to partner or exit to pharma in late stage animal or early stage human trials. But to answer your question, I guess my number one priority are my kids, right? And every day I try to find the time to make sure that I'm doing something with them, spending as much time as I can with them. But outside of that, everything, it's like water. It's like a constant hose filling this tortuous canal of life, right? Or tortuous bucket of life. That things will always take up any spare time that I have. So, yeah.
Well, from all of the experience that you've had of going, through these courses, the pitching, the development, the company development, do you find that being in bioscience compared to any other, people going through a similar trajectory offers a wider range of opportunities, or do you think that this is a time for putting all of those tools to use across physics, chemistry, sociology? I suppose, is biology the best place to be starting, or do you look at chemists with envy and think, oh, they get to do copper nanospheres or something?
Yeah. Honestly, I think one should challenge themselves to study the most rigorous aspects of whatever discipline they're interested in, because fundamentally, if you can learn how to learn, which is a big part of doing a PhD, right? It's learning how to problem solve, it's learning how to learn, learning how to ask questions. If you can learn how to learn, you can learn anything. You can learn anything and learn anything well. Like, for example, to treat patients, one could go through medical school, do their residency, become an attending, and then eventually do a fellowship or whatever, and then do many, many years of training. Or one could become a nurse after 2 years of training, or a PA or a physician assistant, or a variety of other things that require a lot less schooling, but still have a significant amount of patient care, patient-to-patient contact, things like that, right? It may not be the same intricacy, it may not be the exact same thing, but still, if your goal is to treat patients. Now, I think there are multiple ways you can go about in getting to these different points. However, I would argue that by and large, many more physicians do research, do clinical trials, do implement new stages and types of innovative care. as opposed to as many PAs or nurses or what have you. In fact, I would argue there's a lot of nurses who do a lot of research. So anyway, the point I'm trying to make is the more education you get, the longer you're in school, the more time you spend trying to problem solve, the better you are when you approach almost any problem, right? And that could be either in the world of medicine, innovation, entrepreneurship, what have you. When I go out and pitch, a lot of entrepreneurs that I see, a lot of really successful folks that I see have very, very, very diverse background. They come from art, they come from medicine, they come from folks like me, scientists, PhDs, or purely from business. And all of them have a deep and intricate knowledge of whatever product they're pitching, because all of them in their respective fields, it could be whatever, are super strong hard workers, experts. They put a lot of time and effort into that to get to that stage.
Okay. Well, with all of the, I suppose, the diverse backgrounds that you're working with, do you find that, well, I'll stick to the way that I phrase this on paper because I don't think I can put it more suitably, is spinning off for everyone? Is commercialization, is development best suited for every avenue of research or every individual researcher?
So I think the answer to that is yes and no. I think every researcher should go through the process of understanding translation, right? Of understanding from start to finish what that entails. Whether you are a clinical trials scientist looking at, dosing a drug, whether you are a chemical engineer designing a pipe for a reactor, right? I think every person should know from start to finish how their product is going to work. Like when I have a student talk to me about an idea, I ask them, what do you envision on the product insert? You know, a piece of paper when you take Tylenol or you buy a drug or anything, it has a product insert, that piece of paper. What do you envision being written on that piece of paper as all the studies that would have been done to approve this drug, right? How is it going to be dosed? Where is it going to be dosed? What's the dosing formulation? You might not know the answer to all of this or any of this, but at least you should think about it. Understand that this might be important one day. Like you could create a really, really good cure for something, but if it's extremely unstable, you may never get it to the masses. You may create a vaccine for X, but if it can't survive the cold chain to be delivered in remote locations, it may never get there, right? So there are so many considerations that I think every scientist should know about. So in a sense, is translation for everyone? Yes. Knowing the steps about it, knowing the pathway forward for it is critical and essential and should be taught at every level of school, right? Entrepreneurship and translation of ideas, right? How do you take that idea for a lemonade stand and actually implement it, right? There's a Home Depot trip. There's a trip to the grocery store. There is a setup fee. There is a weather check, right? There are so many different aspects and anybody can go into this translational process and gain an appreciation for this. From a lemonade stand to drug development, I honestly believe everyone should plan, right? Yeah. And the no part of this is I myself am not the right person to take my product to human beings. I will tell you that right now. I'll tell you that on every podcast I do until I am, maybe, but I don't think I'm the right person. I think the right person to do this is someone who has raised $50 million with pharma before. I haven't, right? Because at the right time, you need to bring on the skills for that position, right? When we take our product into phase 1 clinical trials, we need to hire a CEO who has done phase 1 clinical trials, who has experience doing that. Then we might need to hire a new CEO or maybe have that person, right, raise a Series A, Series B, have that person exit the pharma, right? A person with track record. If you're going into war, you want to be prepared with the right generals, with the right equipment. Can't go in there not unprepared. How about that? Too many examples to use, but I won't. I think, having that adequate level of preparation is important. So yes, in a sense, I think even I myself, there is a limit to how much I can translate comfortably with efficiency. And after that, it is inefficient and a waste of your time and your innovation to not hand over the reins, not even hand over the reins, but not bring on the right partners at the right time to move things forward. I think that's critical for translation, the team.
If there is a undergrad listening to this who thinks, I just want to do my narrow band of biological research, I want to get into a lab, I want to be there with a pipette for however long that takes, I want to do that and kind of stay in my lane. Do you think that there is going to be a future for keeping the shutters up?
100%. Oh my God. So 100%. And not just that, whatever you are interested in. If you like pipetting, but you go home every day and you just love coding, right? Oh, no, forget that. You love playing computer games, right? or maybe designing stuff, right? Or you like artistic design, synergize those. We live in the age where diversity of thought is celebrated, right? If you can bring together art and medicine and engineering, you are someone that is unique more so than that person who knows only engineering or that person who knows only medicine or something else. Synergizing your talents is important. Ditzing around and wasting time is not, right? Everything you do should be worth something. What is the goal? What are you trying to get out of it, right? Like, what is the win here? What is the achievement? What is the milestone? What is the badge that you're going to have having done this, right? What is the achievement? But if you pipette all day, you go home, you're doing some artistic design, maybe tomorrow you're going to design a new pipetter. Maybe tomorrow you're going to innovate, right? So what I would advise that person who likes pipetting is find whatever you're interested in and deep dive into that and explore, right? Find as many people you can talk to about it, start innovating, think about anything that's innovative and exciting about it, because there are opportunities to innovate. There is money out there for you to build your ideas. And if you have any questions about those things, contact people like me, contact your local entrepreneurship office, right? I don't know if I can make a plug here for people, but the National Science Foundation runs this program called NSF, right? National Science Foundation, NSF, I-CORPS, I-CORP. They call it I-COR, right? I-COR. It is a phenomenal program. It's very, very intense. They can get a little mean at times to make sure you complete everything on time. But if you go through the I-COR program that's available at your local universities, many universities have site programs. And if you're good, you can go to the national program. They will give you money to go speak to people to figure out if your idea is a good idea or a bad idea. Now, obviously, before you do the program, you kind of have to do some homework, your Google searches, things like that. But there are so many resources available. Wikipedia is available, Google Scholar, and then get deeper, get more specific, learn a little bit more and verify your sources, right? But the information is available, verify your ideas, try to innovate in whatever sphere you're in, because that's where it comes from, right? Because if you're pipetting every day, you're going to say to yourself, man, I really don't like X. And that's a gap maybe no one has thought about. Innovate there and make a new pipetter, make a new, what's it called? I don't know, maybe a finger splint, something. There are so many things you can innovate. And just because someone like me sitting in the professor chair barely pipettes, I'd never know about that. I'd miss that. You wouldn't.
Are there any other things to plug? Is there anything else on the near future horizon for what the lab's working on, or any courses that you might be leading at NGIT?
So in terms of NGIT, in terms of education, in terms of courses in general, I would strongly recommend getting into artificial intelligence, machine learning, and quantum computing. I think the future is going to leverage these tools. And if you can speak some of that language, no pun intended with machine learning and AI, but if you can speak any of these languages, I think you will have a leg up. That being said, you can learn a lot of this on YouTube. One of the core languages that a lot of these programs use is Python. You can learn a lot of Python for free on YouTube, or you can learn CS 101, right? You can learn it in a course. But get in there, start learning. In fact, a lot of these resources are available for free from Google or MIT OpenCourseWare, so many different places. Get lost in education, get lost in learning about these things, because I didn't go to school for these things. And I talk about it now because I got lost in it and I just loved it. And I thought, how can I do what I'm doing now, which is peptides, right? Making peptides, making proteins. And how can I synergize that with this really cool technology that's out there right now? And now we have people working on it. So, and speak to people. And the most important thing you can do is stand on the shoulders of giants, right? When you go to Google Scholar on the bottom, below the search bar, it says standing on the shoulders of giants. The research is done by not inventing something out of the blue, in my opinion. Research is done by taking a look at all the work that's been done out there, this huge foundation and body of work, and then saying, what is a gap that exists and how can I innovate there, right? So you have these light posts, and sometimes they become beacons, and sometimes they revolutionize the field. But oftentimes you have this huge body of work and you're making, small beacons of change, innovations.
You say one of the best things you can do is talk to people. If anyone wants to talk to you, where can they find you, either online, social media? What would be the best way to reach out?
So I'm pretty big on LinkedIn. I spend a lot of time there. I guess if I can plug things, I'll plug LinkedIn as well, because I think it's a phenomenal social networking app. Ever since COVID, I have used LinkedIn to make so many collaborations that have significantly changed the type of work I do and the folks I speak to, and the nature of the work I do. In addition to that, it's a great way to communicate with people who you might never have spoken to before. One of the things I've always believed in is people like me, as is evidence right here, love to talk about themselves, love to talk about what they do. So if you email someone or message someone and say, hey, I'm really interested in self-assembly. I'm really interested in how we can leverage quantum computing in peptide design. I'm really interested in, diabetic retinopathy. I'm really interested in therapeutic angiogenesis, dental pulp regeneration, something, right? I'd love to talk to you about it. So reach out with something you're interested in. It always helps to, Google search a little bit, watch a couple of YouTube videos, get yourself up to speed, make the most use of every opportunity. But these conversations will change your life. You learn lessons that you otherwise wouldn't have learned. Also, another thing I can tell you is don't go in as a salesman, right? Don't go in pitching, here is my product, drink it, right? Don't go in saying, hey, I want to be an astrophysicist. Tell, what are the steps I need? I would go in saying, hey, you're an astrophysicist. What do you do every day? What's your favorite part about your job, right? Like discover something true about it. Discover something unique about it. Because everything else you will get from Google, right? Information's out there. But that perspective, that feel, that unique aspect, that's hard, right? And even your chat GPTs are now going to tell you, yeah, I really don't want to bother my son to take me to the clinic. That's a big inconvenience, right? You can ask AI, what's the biggest challenge with developing drugs for blindness? And it might tell you extended release, blah, 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 right? Or efficacy targeting different, soluble VEGF, a number of different things. But fundamentally, through customer discovery, through speaking to people, you're going to realize, that inconvenience should influence how you make things.
I think that it's heartening that for all of our talk about, the cutting edge of technology and the atomic scale resolution technology that you use to examine molecules that go into medicine, that it still comes back to people.
I think it starts and ends with people, right? And my argument is at every step of the way, you should be thinking about those people, whether it's the patients, right? But you can think about that patient, but if the pharma is not interested, they're not going to pick up your product. The patent's going to expire. It's never going to make it to the clinic, right? So you got to think about pharma. You can't think about pharma unless you think about the physician, because the physician needs to have uptake. Otherwise, pharma isn't going to buy in, right? And then you got to think about the insurance company, because if the insurance company doesn't have a reimbursement code for this product, you're going to have to apply for one. It's going to cost several millions, delay your FDA filing, right? So long story short, you got to learn the ecosystem. Whatever game you want to play, right? Go learn the rules, go watch a couple of games, go speak to some of the players, speak to the umpires, speak to the fieldsman, speak to the vending guy, because that vending guy is going to have some lessons that you will never have heard of before. Speak to that who cleans the seats, because he's going to tell you something that you will not know, and then buy a baseball team. You know what I mean? That's the homework you want to do. Before you become a baseball player, go do that. Yeah, doing your homework is critical. It is critical. I would advise every kid who's in high school, in college, who's a professor, right? I would advise you, go out there and speak to people, because I myself and my colleagues make a mistake all the time, make the same mistake all the time, which is getting siloed into this small world of self-assembling peptides and not doing anything else, right? And not exploring, being resistant to changing out of that. And I would argue that unless you capitalize on the latest technologies, you're going to become outdated and you're not going to be innovative. You're not going to change the world. And if you're not doing that, do something else, right? That's what we're here for. That's what's fun. That's the goal of this, in my opinion.
Well, I can't think of any better note to wrap things up on than go out there and change the world.
Change the world. My PhD advisor, Elliot Chaikov, he gave a talk once. And on this last slide, I don't remember the exact words, but, he said, sometimes it gets tiring, right? You're in the lab, you're pipetting, you're doing this animal work, you're doing whatever, it gets tiring. And you're like, you know what? Forget it. I'm going to come back tomorrow. Or you got a time point of the week and you're like, forget it. I'll just do it on Monday. But just keep in mind that extra second that you put in, that extra minute, that extra day, that extra hour, that extra day, extra year, right? For some people, right? That you put in might change someone's life, right? The clock is ticking. The clock is ticking. And ever since then, remember that talk from 2011 at a Gordon Research Conference in Tissue Engineering and Biomaterials in 2011 in June. Till now, the clock is ticking, right? Patients are waiting, the clock is ticking. And that's what drives me. And that should drive all the people in my shoes.